Hello, everybody, and welcome to this week's episode from Bite-Sized Biocompatibility, talking about sensitization. We hope you are sensitive and ready for this one.
No, don't be too sensitive.
Don't be too sensitive about this one. All right, so today we're going to be talking about the overview of the different methodologies for investigating sensitization, where it stands, where the acceptance of each one stands right now, and discussing also how to approach sensitization and evaluating that endpoint.
Yeah, so a lot to cover, a lot of great things around rather historical test, and a lot of exciting news on changes and things that are coming in medical device. So hopefully everyone is aptly hungry for this episode.
Yes, bring your appetites. Hello and welcome to Bite-Sized Biocompatibility, the educational series focused on making the technical content of medical device biocompatibility digestible. My name is Leah Williams.
Hi, and I'm Beau Rollins with Intrinsic Medical Group, and we're excited to take you along on this journey.
Hello and welcome back to another episode of Bite-Sized Biocompatibility, this time episode number 8, and we are talking about all things sensitization.
No irritation puns this time, unfortunately. Sensitization puns don't quite work the same way.
Well, you can definitely make enough comments about being sensitive about things.
Yeah, that is true. That is true. And I mean, as we get into sensitization, it's all about repeat exposure. So, I mean, perhaps only someone can handle so much exposure from myself.
Exactly. Well, hopefully this one doesn't cause a
sensitization response.
It's an adverse systemic response.
Yes.
So, yeah, stay with us. Although by this time you would be sensitized.
Yes.
Anyway, let's get into it before we get carried away in this regard. So, what is sensitization? So from what I'd understood with sensitization is it's a physiological response that after an initial exposure to a certain constituent or chemistry, your body then learns that is potentially a hazardous substance to your body and will undergo a full systemic immune response in response to that. How many times can I say response?
No.
In response to that.
You did great. And this is one of them that, there's a lot of confusion sometimes on what irritation is versus sensitization.
Yeah.
Because they're both measured the same way in the test through redness and swelling. They use the same scale and grading aspect. But you said it eloquently in that sensitization is a systemic response, where irritation, as we talked about in the previous episode, is a local response, right?
Right. And this is the main difference between the two.
Correct. And a lot of times we'll use the example of allergies to people because they understand that. But the concept behind it is that you get exposed to a compound, and that triggers within your immune system and through different pathways, I can't even say the word. It's one of those words that I'm just never going to be able to say properly. Can it?
I don't know what you're after.
I know it's a skin. Can it? You're going to kill me now. Anyway,
right,
we'll cut that bit out because I can't say it right. It's not coming out my tongue.
Yeah, thanks.
But yeah, so there's physiological pathways that respond to that chemical compound, and they create a memory. So as part of that, like the first time you get exposed, nothing happens.
Right.
Generally speaking, you wouldn't know that you were exposed, but your body does, and it creates a memory.
I really like that word, that use of the memory from that. It really, to me, helps define exactly what sensitization is.
And different than irritation, right? Where irritation, we put something on and it's going to cause a response at that site. After you've got that memory, you could get exposed anywhere else and you'll cause a response. And it generally is an immune response, and it can be systemic. So it means it can have a cascading effect depending on the exposure and the amounts. So that's where a much larger risk factor or harm factor comes into play with sensitization.
And that's where you see something like with that cascade effect, say somebody has a peanut allergy and you see them having a full anaphylactic shock, for example, and that would be an example of how a very small concentration of something can really compound into this full-scale systemic response.
Yes. And so as you can imagine, doing this test is rather challenging, or can be, just by the nature of having to expose animals to do induction phases and then go through the exposure phases. There actually is a part, the first part, where it's measured almost like an irritancy, because we don't want an irritant to give us a false sensitization read, right?
Right, especially if we're still using those measures of swelling and redness as the
Correct.
indication for
Yeah.
a degree of sensitization.
Which, on a side note, is one I've always wondered, why can't we use sensitization as also an irritation test? I'll just
Well, why can't we?
throw that out there. It's a good question, because if you are checking for irritation and you don't see any redness or swelling as
Yeah.
part of that time point, why couldn't you just use that test?
So yourself, Beau,
I
who is
know.
working on Working Group 8, along with the other members in Working Group 8 who cover both irritation and sensitization. If anyone's listening out there and wants a project, please, you're welcome for the idea.
Yeah. So it's definitely one we'll continue to discuss, being able to consolidate. But as of now, they're two separate tests. And then an interesting little side fact that sometimes you'll get questions on is the controls that are used for the sensitization test. I've had several times where the FDA has asked for, like, you'll get a report, and most labs won't run a positive control with the study
Wait, why is that?
because they're extremely dangerous compounds
Substances,
and
right?
substances.
So you don't want to stock them in the lab.
For employee and health and safety, we don't want people contacting sensitizers on a routine basis.
True. That could be problematic
Yeah.
and probably result in some sort of lawsuit or a lack of lab
Just-
technicians working on it.
Yeah, just health and safety. So
Yeah.
most labs will set up and run on a quarterly or semi-annual basis, where they'll still, I mean, it's kind of silly that we do that anyway. These are well characterized already. We know that they're sensitizers. We shouldn't need to verify that this works on an animal every time. But nonetheless, that is still something that's pushed through. But that will come up as a question, because most time, if you look at your report, you'll notice that a control was not run, a negative, just to verify that
Yeah.
there's nothing wrong with the solvents in that are utilized. But there's not going to be a positive, and there'll be usually reference to another study that was done for the positive aspect.
Yeah. So going a little bit further into that, then you started talking a little bit about the studies, and you've mentioned an animal study there. So could you just give me an overview of the in vivo method and how it would typically look to conduct for sensitization?
Similar to the irritation, we have two in vivo models. So we have the Buehler method, which will be a primary skin type contact. And then we have the guinea pig maximization. Those are your primary in vivo. And then we also have LLNA, the local lymph node assay. By far, the guinea pig maximization is going to be the gold standard. It's primarily the one that has the longest running. It's been utilized for decades, right?
And my understanding is that that Buehler method is a direct contact method.
Yes.
And then the guinea pig maximization test and the LLNA method are extract-based methods.
Yes.
Okay.
LLNA occasionally has shown in the past some interferences with certain metals and things. Overall, it's a generally good assay, but as far as like the agency's concerned in that, they're much more comfortable with the maximization. So most people go to the guinea pig as far as the model of choice or the method of choice when they go to evaluate sensitization.
It's familiar, right? And so often we see this is a big frustration that I personally have within the industry, is that that's how we've always done it. So therefore, that's what we stick with a lot of the time. And it makes it very challenging when new scientifically accurate methods are coming forward that have been demonstrated to actually be maybe better or more validated as well than the existing historical methods.
Yeah.
Talking of which, I believe there's also an in vitro method for sensitization.
There is. So right now is a very hot topic time, I guess, for sensitization. As we've been trying to get irritation, and we discussed this in the last episode on in vitro irritation, we've been trying to get that wrapped up and globally accepted. So there's been a little delay on the sensitization, but as far as the rest of the world, right, with pharmaceuticals, cosmetics, and all that, they've been using in vitro sensitization for a long time now. there's a lot of OECD guidelines out there on in vitro sensitization, and there are a few key players with Sensigen, Episkin, Matec. They're all developing in vitro assays and protocols that are more specific around medical devices, with the intent and purpose of setting up and validating.
Right. And my understanding as well from these methods is that they have been demonstrated to be much more accurate and reliable in identifying sensitizers as well.
Yes, that's a fair statement. So the guinea pig is ironic because there was a movement a while back where Nelson Labs went and pulled a bunch of sensitization data, and then they started reaching out to others like at NAMSA and WuXi and others.
We're talking
LabCorp.
like thousands of data points. So.
Thousands of data points
Yeah.
on sensitization to be able to look and find that there was, I believe, one case, but when further reviewed, it ended up being determined that it wasn't. So out of tens of thousands of sensitization tests that have been performed on guinea pigs, we're not actually seeing...
We've just seen one failure, which was determined that it wasn't actually down to sensitization.
I believe so. On that one, obviously failure data is always a tricky one, but regardless of if we count it as a true failure, we're still talking about literally tens of thousands of these studies on medical devices and not a single sensitizers because they're well known and we don't manufacture with them in there.
Exactly. So that was going to be my follow-up question actually with that is, to what degree could this just be that, we don't intentionally manufacture with chemistries that we know cause sensitization responses?
Every manufacturer in medical device has safeguards in place against dangerous and caustic chemicals. You can't ship things around without clear indications and controls. They can't be received without those as well. So the likelihood of one of these sensitizers getting in is very low, as we've seen, because we've not picked it up on any sort of routine testing and things.
Right.
And even with MDR and some stuff where we've gone back and retested these devices that have been out forever and all these tens of thousands of tests, we're not seeing it.
Yeah.
So the overall risk of finding it in our industry is incredibly low. Yes, the risk to patients is high if they got a sensitizer. And I think that's where we talked earlier about risk, about that probability and severity. High severity, very low probability, which should drive on that calculation to be a low risk that maybe doesn't need...
Or at least a medium risk, right?
Yes.
Or at most a medium risk, I should say there, yeah.
So when we talk about the in vitro methods, since you brought that up as well, there are a couple criteria around OECD. There's like 5 metabolic pathways. We're not going to get into the detailed science behind sensitization on here, but there are requirements around these in vitros that they satisfy a couple of these pathways, and that's been kind of the challenge on developing these and verifying and validating these that they can satisfy this requirement. But we are, as far as Working Group 8 is concerned, at the beginning phases now of detailing and building out the validation plan for a variety of these models so that we can do similar to with in vitro irritation, where we can sponsor and execute a proper test method validation that can further support, hopefully, the acceptance into industry.
Yeah, that sounds fantastic. So we've looked a little bit into kind of like what's on the horizon, the future of these in vitro methods and all of that. I'm actually curious to know a little bit more about the history, because prior to the 2023 update, -10, so irritation, and -21, sensitization, were combined.
23.
It was a 2021. Okay. Prior to the 2021 update of the standard, -10 for irritation and -23 for sensitization were combined within one standard. So just talk me through a little bit more. What was the rationale behind splitting those out?
So,
and why did they end up together in the first place?
Yeah. I think because the reading is the same, meaning the scale and looking on that. So in the standard itself, there is actually a picture on reading that's redness and swelling and how to list it. Because they use the same scale and the same grading criteria, they ultimately were kind of grouped together and sensitization utilizes irritation at the very beginning read point. They were combined in 10 at the very beginning. And then when we started to develop and validate an in vitro method, it was Working Group 8's desire and feeling that now is the time to split this, because we're going to be developing in vitros for both of these, and they're going to be much more unique in this regard, not being similar at all.
Less overlap in the methodology.
So the decision was at the time, because in vitro was coming on and getting validated, that we'd create 23 and move irritation into 23. And that way it can be focused on it primarily, like listing and incorporating all the in vitro aspects. And then -10 was basically stripped to just contain sensitization. And there was wording put in, and there's now a TS that has also been released that...
A technical standard.
Yes, a technical standard that explains how to validate a sensitization in vitro assay. So all the framework and foundations being laid to basically allow for this going forward, but it has created now two separate standards for industry to use, and I think that ultimately plays well to the future of this, to allow that specificity for those different assays.
Yeah, for sure. Yeah, so from what I've understand, then the current state of the industry in terms of developing that in vitro method is that whilst maybe it was put on hold a little bit more for whilst pushing that in irritation in vitro method forward, it's now kind of going full speed ahead again and trying to get it fully validated and ready to go pretty much as soon as the irritation is.
Yes. There's several models already validated for OECD, and that was kind of a lot of those work for Sensigen and Episkin and Matek and all that. So they've gone through and done their validations with OECD and acceptance there. And now it's same kind of challenge that we had with irritation is we have to create a protocol that is appropriate for medical devices with an extraction of a device and a sensitivity with the assay that's appropriate. I think this is a more challenging one because we don't have any animal data that really shows the response on things. So a lot of this goes off of historical data that exists in that comparison. But oddly enough, a lot of us feel that in vitro sensitization is going to be more likely accepted than the in vitro irritation.
Why do you think that is? Is it just because we have that lack of data demonstrating in the in vivo models that it can maybe be protective enough to demonstrate? Is that the argument and rationale, whether it's right or wrong?
Believe it or not, even though sensitization is a more complex response, it's very well characterized, and there's certain pathways, as mentioned. And as long as
comes with the higher risk of the response, right? Yeah.
And the repeat dosing, I think, and then looking at the response, right, basically kind of amps up that risk or mitigating that risk aspect of missing weak sensitizers.
Right.
And a lot of there's not quite the same as weak sensitizers as there is with weak irritants. And ultimately, that's kind of the concern on the in vitro model, is that it's not going to be accurately depict those weak sensitizers and mixed sensitizers or irritants. I'm so sorry that the irritation will not pick up the weak irritants, but that with sensitization, because it's a cascading systemic effect, you will easily pick it up.
Makes sense. So that's sounding really quite optimistic for the future there. I think just to wrap it up, let's kind of end with a question of, unless you had something else to say.
No.
But yeah, let's end with the question of, for somebody who's coming into doing a sensitization study, you've identified that maybe there's an unknown risk associated with that sensitization biological effect. What are the key things that you need to think about when going into that sensitization study?
So understanding that, one, you're going to be required to do it by most regulators.
It's one of the big three.
Is the first step. So yes, there are theoretical ways to justify out of it, but as of now, and I know some people are going to be able to, but it's not really worth that effort at this point. You still can, as far as practice and taking the correct approach by doing that through your TRA, and determining that risk to be low, but just be aware for regulatory approvals, you're going to still need to perform a sensitization test. At that point, your options are limited at this point in time. And the risk is low at seeing a response with the guinea pig maximization. That overall, that's probably the easiest one or safest one to run.
Regulatory safe, or
just in test. As we talked about, we've not seen any response or failure. So if you're looking at executing a test that gives you, it's already low risk. I'm trying to establish that first, right? We've already determined that there shouldn't be sensitizers there. So this is more of going back to that checkbox. I have to have a test.
Yeah.
And if you're going to execute tests, and you want tests that are going to pass, right? To give you the cleanest results.
Yeah. Unfortunately.
This is the reality, right? Then since the guinea pig maximization is one of your safest bets.
Yeah.
You can go LLNA, but I have run into occasionally issues with LLNA tests with just method incompatibilities and suitabilities and stuff, and less with the guinea pig. So from a risk perspective of execution of tests, maximization's the way I would recommend, right?
Cool.
So, and that kind of goes when we talk about the big three. We talked about cyto and the variations there and getting that right to the device. Irritation, you're pretty much just intracutaneous reactivity, unless you truly have just an intact skin device, which a lot of those you can justify out with the FDA guidance, NXG. And then sensitization's guinea pig. So those are kind of like your big three, your main three. and that's kind of typically ran for most manufacturers.
Yeah, that's really well summarized. Thank you so much for that. And I think just with that as well, I just want to say, just because you maybe aren't able to kind of justify out of doing the sensitization test yet within the current kind of market or the current scope of the field, do keep putting in that literature review, making it very clear and kind of like putting forward through the -18 chemical characterization, especially that data gathering step, that based on the literature, there are no known sensitizers used in the manufacturer as well. So it's just about making sure that we're getting the industry very familiar with seeing that type of input as well.
Yeah. All right. My advice to anyone in biocompatibility in general is that you can either be proactive or reactive.
Yeah.
And the reality is you have to make a decision. When you're putting together your evaluation, your BE, you're either proactive or reactive. And there are a lot of companies and people that choose to be reactive, meaning that they do what is accepted today and that's it. And they don't want to push the envelope. They don't want to start preparing for something new until it's actually in effect. Mm-hmm. And then you get the group of people who want to be proactive, who can see that coming forward, we're going to be utilizing TRAs more in time, right? We're going to be seeing in vitro assays coming in that. So they're already setting up their systems and processes and practices now so that when those things come out, they're already built into the process, which means using your TRA to assess irritation sensitization as part of your process and including that into the biological evaluation. And then that way, when those are allowed, you're already doing it. It's already built in. And even now, if you are doing dual data with like irritation or even with sensitization, yeah, the in vitro may not be accepted at all markets today, but you already are generating that data. You're getting used to that data, interpreting it with your products and understanding it. So that way, when it becomes accepted, you easily phase out the in vivo and you've already got that running and understood.
You're ready, comfortable and ready to go.
Yep.
Yeah. Proactive, not reactive. And that could be good advice for life as well. So
Yes.
All right. Well, I think that just about brings us to time. So thank you, everybody, for joining us for another episode of Bite-Sized Biocompatibility. We hope you enjoyed this. And whilst you're digesting, as per usual, feel free to reach out to myself or Beau via LinkedIn or our emails, which we'll drop in the caption below. Until next time.